Hello, welcome to Study G. Today we are going to be completing the chapter Microbes in Welfare. Let's begin. So what are actually microbes? What type of organism comes under microbes? Bacteria, fungi, protozoans, viruses, viroids, prions. So these are the organisms which come under microbes. Now, where do these microbes live? So now coming to the location, they are found in water, air, soil. They also live in or on animal/plant bodies. In special cases, they live in highly acidic environment as well. So, the bacteria which live in highly acidic environment are called acidophiles. These also live in thermal vents. Thermal vents are geysers where the temperature is greater than 100 degrees Celsius. So in this temperature, no other organism can survive except a few microbes. So these microbes are usually called thermophiles. Now we are looking at the diagram. So this is a bacterial colony which is growing on a petri dish, and this is a fungal colony. Next we have the different types of bacteria. So this is the rod shaped bacteria. The second one is a coccus, or also called as spherical shaped bacteria. There is third one, a rod shaped bacterium with flagella. So the flagella usually helps in the movement. Next we learn about virus. So bacteriophages are the viruses which feed on bacteria. So they feed on bacteria. Now coming to adenovirus. So they are responsible for causing respiratory infections in humans. Now coming to tobacco mosaic virus, or TMV, they are compact rod shaped viruses. Coming to curd, so the bacteria which is present in curd is called as lactobacillus. So this lactobacillus is also called as LAB, which is lactic acid bacteria. So they are called so because they usually produce lactic acid after the partial digestion of milk protein. We'll learn that in detail now. So what usually happens during the formation of curd from milk? We'll note that down. So what happens? You take milk and curd. So you add inoculum. Inoculum is also known as a starter. So these starters contain millions of LAB which multiply in the curd. Okay, now the milk is converted to curd by adding inoculum. So what happens is, when we add this inoculum at a particular suitable temperature, these inoculums multiply. So we can write it here: starters multiply at suitable temperature. So this is all about inoculum. So inoculum is LAB only. So what actually happens is the partial digestion. So partial digestion of milk proteins by the lab gives curd. Suitable temperatures, because usually in summers we can set the curd faster compared to in winters. So suitable temperature is also required for the multiplication of the bacteria. So during this process, during this conversion or the digestion, lactic acid is produced. So hence they are called as lactic acid bacteria. Now coming to the nutrition. Coming to the nutrition of the curd. Curd is usually more nutritious than milk. So this point you need to know. Curd is more nutritious than milk because of the presence of vitamin B12 in an increased amount. So also this LAB keeps check on disease causing microbes. In stomach. All about curd. Now coming to the fermented products, dosa, bread and toddy. All these three products which we use in our daily lives are fermented through one or the other means. In the dosa again, CO2 is released by the bacteria. Naturally bacteria is present in the rice and the lentils, so fermentation of batter from bacteria. So during cooking what happens, CO2 is released, which gives it a fluffy or a puffy appearance, Now coming to the bread, the organism which is used in bread is called as Saccharomyces cerevisiae. So this is also called as baker's yeast in this case. So what happens again, these baker's yeast or the Saccharomyces cerevisiae, it ferments the dough. So ferments the dough. Again CO2 is released during baking or cooking, during baking, making the bread soft, porous. So this is all about the bread. Now coming to toddy. What is toddy? So actually toddy is a southern Indian traditional drink which is alcoholic drink. It will be a liquid which is a white in color, and again it is fermented. So here what they do, how they form the toddy is from the sap of palm. The sap of palm is fermented to yield toddy. So the naturally present yeast ferment toddy and makes it alcoholic. Now coming to the next part, we have here about cheese. Actually, cheese is one of the oldest food where microbes are used. So also another point is that we obtain different types of cheese depending on the type of microbe which we use. So for example, if we use this bacteria, that is Propionibacterium shermani, we get Swiss cheese. If we use Penicillium roqueforti, we get Roquefort cheese. Similarly, we use different types of microbes to get different types of cheese with different taste and texture. So the taste and the texture also differs depending on the microbes which we use. So now coming to the first, Swiss cheese. Propionibacterium shermani is used to ripen the cheese. So here we could see big holes are formed. So this is formed due to CO2 release. So carbon dioxide is released, due to which you get that big hole-like appearances in the cheese. This is Roquefort cheese. This is again fermented by Penicillium roqueforti. This is a fungi. One is a bacteria. Now coming to this Camembert cheese, again this is also ripened by a fungi. This is called as Penicillium camemberti. So this is all about cheese. Now there are other foods also which are fermented, such as soya beans, bamboo shoots, which is common in India as well, and even fishes are also fermented. So soya beans are actually fermented in the Japanese cuisine, where they prepare a dish called natto, which is like a sticky soya beans and is usually consumed with rice. The organism which we use is again called as Saccharomyces cerevisiae. In this case it is called as brewer's yeast, or you can also call it baker's yeast. Both are one and the same. So we use fermenters, which are large vessels used to ferment alcoholic beverages on an industrial scale. Large tank. So how are the alcoholic beverages produced? So the yeast, what it does is it ferments. The yeast ferments fruit juices and cereals to produce ethanol. The substrate which is used in alcohol industry is called as molasses. So what actually happens? The yeast feeds upon the cereals or the fruit juices, and through anaerobic respiration it forms alcohol. So through anaerobic respiration it forms alcohol. Now here we see, depending upon the type of raw materials used for fermentation and the type of processing, we get various types of alcohol. For example, with the raw material, the beer is prepared from barley malt, while the wine is prepared from fruit juices. Whiskey is prepared from fermented cereals. Brandy is again fruit juices are used. Rum, we use molasses to prepare rum. And gin, we use rye malt. And for vodka we use potatoes. And another interesting thing we should note is the type of processing. So the type of processing also yields different type of alcoholic drink. For example, here the beer and the wine, these two are non-distilled, hence they have very lower concentration of alcohol. While here, when we compare it to whiskey, brandy, rum, gin and vodka, the alcohol percentage is very high. This is because they are distilled. Let's see the types of processing. So without distillation, we get lower alcohol concentration. Examples can be beer and wine. And with distillation, we get higher alcohol concentration. Example, we have brandy, whiskey and rum, etc. So these are the main examples which we need to remember. Coming to the next part, we have about antibiotics. So the chemicals having the ability to kill or retard the growth of a disease causing microbe. These chemicals are known as antibiotics. So the first antibiotic which was discovered was penicillin, and the organism from which we got penicillin was Penicillium notatum. Penicillium notatum, it is a fungus. Alexander Fleming was working with a bacteria called as Staphylococci bacteria. So what happened is, in one of the unwashed culture plate, there was a mold which was growing, that is the fungus which was growing. So wherever the fungus was growing, the bacteria could not grow. So he observed that there was some sort of a chemical compound which was released by the fungus that did not allow the growth of the bacteria. So then he named that compound as antibiotics. But however, what happened that he could not completely establish a fully effective antibiotic, and it was done by other two scientists. Their name was Ernest Chain and Howard Florey. They showed full potential of an antibiotic. Unfortunately, Alexander Fleming died before they could show the full potential of antibiotics, and later these two scientists, that is Ernest Chain and Howard Florey, they together showed the full potential of antibiotics. Hence all of these three scientists were awarded Nobel Prize in 1945. Fleming, Ernest Chain and Florey. So these three of them together got a Nobel Prize in the year 1945. So why did they use this antibiotics? So antibiotics were used to treat soldiers, were used to treat American soldiers during World War II. Antibiotics are used to treat plague, whooping cough are also called as Kali Khansi, diphtheria, that is Galghotu, and leprosy, the Kustrog. Now coming to chemicals, enzymes and other bioactive molecules. So here we have organic acids. So the organism which we use to produce citric acid is Aspergillus niger. Acetic acid is prepared from the bacterium called Acetobacter aceti. Butyric acid is prepared from the bacterium called Clostridium butylicum. And lactic acid, as we all know, lactobacillus is used to produce lactic acid. So in this case, this is a fungi and the rest three are bacteria. Now for alcohol we use again Saccharomyces cerevisiae is a yeast in the commercial production of alcohol or ethanol. Commercial production ethanol. Now coming to lipases, the organism which we use to prepare lipase is called as Candida lipolytica. And the use of lipase, we usually use it in the detergents which remove oily stains from clothes. So lipases are those substances which we use to remove oily stains from the laundry. So this is all about lipase. Now coming to streptokinase. Streptokinase is extracted from a bacterium called Streptococcus. So the main use of streptokinase is as a clot buster. So this clot buster removes blood clots from patients who have undergone myocardial infarction, that is heart attack. So to remove that particular blood clot from the blood vessels, they provide streptokinase to the patients. Next we have about cyclosporin A. Again we use a fungi in this case, that is we use Trichoderma polysporum. Is a fungi. Now the cyclosporin A is used as an immunosuppressive drug. So immunosuppressive drugs are those drugs which are provided to the patients who have undergone organ implants. So to prevent the immune system from attacking those grafts. Now coming to statins. Use Monascus purpuris. Is a kind of yeast. Monascus purpuris is an yeast. So again it is used as, so statins are used as commercial blood cholesterol lowering agent. So what the statins does is it acts on the enzyme responsible for the synthesis of cholesterol. And prevents the formation of cholesterol. Next we have about pectinases and proteases. So the organisms which produce these are Aspergillus niger. Aspergillus niger is responsible for producing pectinases, while Bacillus species are responsible for producing proteases. So the use of pectinases and proteases is to clarify the fruit juices. Now if you compare the homemade fruit juices to the ready-made bottled fruit juices, we find that the fruit juices which we prepare at home are usually not that clearer. It has a lot of fiber in them. When you compare it to the ready-made juice, it is a lot more clearer. So usually in these industries they use pectinases and proteases to clear out the juice and make them a lot more clearer. The major components of wastewater is human excreta, and these excreta usually contain a large amount of organic matter and pathogenic microbes. So hence we cannot directly put this into the water bodies. So hence they need to be processed in such a way that these pathogenic microbes are removed. Coming to the organism there are naturally heterotrophic microbes which are present in the sewage. So we use these microbes to treat the excreta. So there are three stages of sewage treatment. One is primary treatment. Second, we have secondary treatment. Usually, if we are using the water for drinking purposes, we do the tertiary treatment as well. Now coming to the primary treatment. So actually primary treatment involves physical processes only, where we remove large and small particles through filtration and sedimentation. So in the sequential filtration, what happens? We remove floating debris. Remove the floating debris by using fine mesh. So the wastewater is passed through these meshes which filter out the floating debris. Next we have sedimentation. In this, the grit is removed. The grit consists of soil and small pebbles. So these are removed through sedimentation. So the sewage which undergoes these two steps, it is called as primary effluent. So we can write it this way: the sewage undergoes primary treatment to form primary effluent. Now this primary effluent is sent out for secondary treatment. Now coming to the secondary treatment. Secondary treatment is also known as biological treatment. So what actually happens in the secondary treatment is the primary effluent is brought into the aeration tanks, these large aeration tanks, where they are constantly agitated so that the air is passed into the primary effluent. So what this does is it allows the aerobic heterotrophic microbes to grow vigorously. So as mentioned here, primary effluent is passed into large aeration tanks where it is constantly agitated mechanically and air is pumped into it. So what this air does is it provides suitable condition for this aerobic heterotrophic microbes to grow. So flocks are the association between masses of bacteria with the filament of fungi to form mesh-like structure. So as flocks are formed, these flocks, they feed upon the organic matter which is present in the primary effluent. So here in the image we can see this is the aeration tank where the air is being supplied constantly, and this will allow the growth of the heterotrophic microbes. Now what happens? The flocks, they feed on the organic matter. So due to the feeding, what happens? The BOD reduces. So coming to BOD, if we have to define BOD, BOD is biological oxygen demand. So BOD is the amount of oxygen required to digest or oxidize the organic matter in 1 liter of water. So this means the amount of oxygen required to digest or oxidize the organic material in 1 liter of water by the bacteria. So from this what we can assume that if we need more oxygen, that means there is lot of organic matter and hence the water is more polluted. If BOD is high, then the water is polluted. If BOD is low, then the water is less or not polluted. So now what happens, as these frogs feed on this organic matter, the BOD is ultimately getting reduced. Hence the pollution is also reduced. So now what happens, after all these frogs feed up on the complete organic matter, it is sent to the secondary settling chamber. This is the settling chamber where these frogs are allowed to settle. So what happens here is, the frogs are settling here. You can note here, frogs are settled. So these frogs is now called as activated sludge. These are called as activated sludge. So now what happens with these activated sludges, some of the part of the sludge is sent back to use as inoculum. To the next upcoming batch, we add a little bit of this activated sludge so that it could act as the inoculum for the primary effluent which is coming next. Now with the remaining activated sludge, they are sent to anaerobic sludge digester where they are again processed. We will learn that next. So this is all about the frogs. So if we have to define activated sludge, then it would be, so activated sludge is the sediment of the secondary settling tank which consists of bacterial frog. So activated sludge is the sediment of secondary settling tanks consisting of bacterial frogs. So now what happens in this anaerobic sludge digesters? The sludge or the frogs are digested by the anaerobic methanogenic bacteria. As the byproduct of the digestion, we get methane, hydrogen sulfide and carbon dioxide. So if you can remember, these three are the constituent gases present in the biogas. So it can be used as a source of energy. Well, now if you can focus with the figure, the sludge is coming through the inlet and the sludge is being fed by the methanogenic bacteria, and here the gases are rising, that is CH4, carbon dioxide, H2S. These gases are flowing out. The remaining sludge, which is a waste, it is sent through an outlet and it is mostly used as fertilizers in some cases, or it is directly disposed. The water is being used for drinking purposes, then we take it through a tertiary treatment as well. If not, then we will send the water directly to the water bodies after the secondary treatment itself. So this is a physiochemical process involving chlorine gas, ozone gas, zirconium, perchlorate salts, UV rays and sometimes reverse osmosis, that is the RO which we use at our homes. So these processes are done to clear out, usually to remove DDTs, pesticides, pathogens, and some turbidity in the water. Then this water can be used for drinking purposes as well. Now if we had to summarize the whole sewage treatment plant in a single diagram, we could do this. So first, the raw sewage comes through the pipes, that is the inlet. Here it undergoes two types of filtration. Here we use screens, that is sequential filtration. Sequential filtration happens, through which floating debris is removed. Now we have sedimentation here, through which we could remove the grit, that is the soil particles, and a little of pebbles. These are removed through sedimentation. Now this is called as primary effluent. Now it is sent to the aeration tanks. Again, here you can see in the diagram, they are mechanically constantly agitated to produce flocks in this, where the flocks consume the organic matter present in the water and reduces the BOD, that is the biological oxygen demand. See, the air compressor gives out air to the aerobic microbes which digest, and then they are sent to the secondary clarifier where they are allowed to settle. This is called activated sludge. Now some of the part is sent as an inoculum. Here we use it as inoculum. Next, here we do anaerobic sludge treatment. Where both the settled particles from the primary effluent and the secondary effluent are combined in a place and they are treated. Now the secondary effluent is either directly discharged to the surface waters, or it may undergo tertiary treatment if needed. So this is all about the sewage treatment plant. So next we have about river action plans. The Ministry of Environment and Forest has initiated Ganga Action Plan and Yamuna Action Plan to save the major rivers of the country from pollution. So under these plans, what they plan to do is they try to build large number of sewage treatment plants, so that before the sewage is discharged into the rivers, we can treat that sewage and then we can discharge them in the rivers, so that the rivers do not get polluted. So actually the Ganga Action Plan was initiated in 1985. So what is a biogas? Biogas is actually a mixture of gases which is produced by microbial action. So what happens? These gases are produced as a byproduct of the microbial action. So the gas which we get is the byproduct of growth and metabolism. So the gas is a byproduct of growth and metabolism of the microbe. Now, the type of gas which we get usually depends on two conditions. One, the type of microbe, the substrate which we provide the microbe. So these two determines the gas released. So now coming to the main components of biogas, we have majorly methane, that is CH4, carbon dioxide, hydrogen and hydrogen sulfide. So coming to the percentages, usually methane is very high in the biogas and it constitutes about 50 to 70% of the biogas. Carbon dioxide is 30 to 40%. And coming to hydrogen, it's 10%. So coming to the uses of biogas, it is usually used as a fuel in cooking and in lighting. So due to the presence of methane gas, it can be used as a fuel because methane is inflammable gas. Now coming to methanogens, these are the bacteria which feed on cellulose, those to give biogas. So they are usually present in the rumen of cattle and helps in the breakdown of cellulose. So these cattle, the major part of their diet is through the plants, and these plants, they are very high in cellulose. Hence this cellulose is usually broken down by methanogens, and the excreta, through these excreta, these methanogens come out from the cow's rumen, and this excreta, which is highly rich in the methanogen, is used in the biogas plant to produce the biogas. And another point which we should note down is there's a particular question in the NCERT where they have mentioned, can human beings be able to digest cellulose? So one point which you should remember, even in cattle and humans, there are no enzymes present which is responsible for the digestion of cellulose. Both humans and the cattle, they both lack that enzyme. But in cattle, what happened, due to the presence of these methanogens in the rumen, it facilitates the digestion of cellulose. They are also found in anaerobic sludge. The cow dung is rich in methanogen. So now coming to the biogas plant, it consists of main three parts. One is the concrete tank, which is usually 10 to 15 feet in depth, and this is the place where the slurry is fed. So here you can see slurry consists of dung plus water. So here it is a concrete tank which is fed with the slurry. Next there is a floating cover which is placed right over the slurry. So as this floating cover rises, the gases are supplied through outlets. So this is the outlet to the nearby locality. There is also another outlet through which the already used slurry is sent, and that particular waste which is remaining can be used as fertilizers to grow crops in the rural area. As cow dung is available in large quantities in the rural areas, the biogas plants are usually set up there. And also, mainly due to the efforts of the Indian Agricultural Research Institute and the Khadi Village Industry Commissions, the biogas production was developed in India. So what is a biocontrol? So biocontrol refers to the use of biological methods for controlling plant diseases and pests. So usually while growing crops, the crops are more susceptible to the damage from the pest. So what we do is we use that biological, that is a safe method for controlling these diseases and the pests. So why the use of chemicals such as insecticides and pesticides is bad? So one, they're toxic. Another, they are harmful to humans and animals as well. Example, DDT, they pollute the soil. Usually the weedicides pollute the soil, and also it kills both helpful and harmful life forms indiscriminately. So they don't have a target organism, but they just simply kill all those life forms which comes in contact with the particular chemical. Now here we have the approach of the biological farming. So the So what are the approaches of biological farming? Let's see them. 1. Becoming familiar with various life forms that inhabit the field. 2. Becoming familiar with predators and pests and their life cycle. This point is important because if we know their life cycle, then we can find a solution such that we cannot let the life cycle to continue. Next, becoming familiar with the feeding pattern of habitats of pests. Go to the example of biocontrol agents. So here we learn about 5 different types of biocontrol agents. Coming to the number 1, ladybirds. So these ladybirds are used to eradicate aphids. So these are a type of insects which destroy crops. So ladybird is used to get rid of aphids. Next, we use dragonflies to get rid of mosquitoes. Next, we have about Bacillus thuringiensis. It's a type of a bacteria. We use Bacillus thuringiensis or the BT to keep away some certain butterflies and caterpillars. Next, fungus trichoderma is again we use this to eradicate various pathogens. And baculoviruses we use to get rid of insects and certain arthropods. Now let's learn about the Bt crops in detail. So actually Bt is a type of a bacteria. So these bacteria are available as spores which is mixed with water and then it's sprayed on these plants. And the predator which we target is the insect larvae such as caterpillars. So what happens when this caterpillar feeds on the particular Bt spores, the spores go to the gut of this caterpillar where a certain type of toxin is released and it will cause lysis in the gut, that is a cell death, leading to the ultimate death of the caterpillar. We'll learn about this particular Bt crop in detail in the chapter biotechnology. Toxin is released leading to the death of the larvae. Another interesting point is does not affect other insects. So it only affects larvae such as caterpillar. So transgenic plants are developed through genetic engineering having Bt toxin gene. So what happens in these plants is these plants become resistant. So these plants become resistant to insect pest. So a gene is inserted. So this gene, what it does, it releases the same toxin which is responsible for the death of the larvae. Example of this transgenic plant is Bt cotton. Next we have about the fungus trichoderma. This fungus trichoderma is a free living fungi and it's mostly found in the root ecosystems, and these are effective against several plant pathogens. Now coming to baculoviruses. Baculoviruses are the pathogens that attack insects and other arthropods. So coming to the example, usually the baculovirus from the genus Nucleopolyhedrovirus. These are used to get rid of the pests. So the advantages of using baculovirus are they are species specific and they have a very narrow spectrum insecticidal application. That means they only target a very narrow amount of organisms. So another point is they have no negative impact on plants, birds, mammals, fishes and even non-target insects. So the narrow group of the insects, only these are affected by these baculoviruses. So this point, that is no negative impact. So this is an important point in the integrated pest management program, because in this integrated pest management we usually focuses that all other organisms which affect the plants are not is used when treating an ecologically sensitive area. So IPM is an ecosystem based strategy that focuses on long-term prevention of pests or damage caused to crops by combination techniques, such as in this integrated pest management we use biological control, habitat manipulation, we use resistant varieties of the crops, modification of cultural practices to give better protection to the plants from the pests. So organic farming is the production of crops through the use of biofertilizers, as they provide optimum nutrients to crop plants. Another feature of this biofertilizer is they do not pollute the environment or do not cause any problems to the organisms living around. So production of crops through the use of biofertilizers. So what are biofertilizers? Biofertilizers are the organisms that enrich the nutrient quality of soil. So what these organisms do, that they either provide nutrients such as nitrogen, phosphorus, water and minerals in an easily absorbable form to the plant. So the main source of biofertilizers are bacteria, fungi and cyanobacteria. So coming to bacteria, rhizobium, so it forms symbiotic association with the root nodules of the legumes. Symbiotic association, and fixes atmospheric nitrogen in organic forms such as ammonia, that is NH3, nitrites and nitrates. So what this rhizobium does is it converts the atmospheric nitrogen into organic forms such as ammonia, nitrate and nitrites, because these are easily absorbable forms of nitrogen. Now coming to free living bacteriums, the examples include azospirillum and azotobacter. Again, these also fix atmospheric nitrogen to the soil. Hence this increases the amount of nitrogen content in the soil. Hence again it helps the plant to easily get nitrogen from the soil. Next we have fungi. So mycorrhiza is the symbiotic association with fungi and roots of the plant. So usually the members from the genus Glomus form mycorrhiza. So from the genus Glomus they form mycorrhiza. What this mycorrhiza does is it provides phosphorus. So it provides phosphorus to the plant. through soil. So as this is related to the plant, so the plants which have mycorrhizal association show specific type of features such as resistance to root borne diseases, tolerance to salinity and drought, and overall increase in plant growth and development. So the fungus helps in these three features for the plant. In return, the fungus gets the food and shelter from the plant. Now there are two types of mycorrhizae. So now coming to ectotrophic mycorrhizae, they form mantle on the outer surface of the root. So it helps in the absorption of H2O and minerals such as nitrogen, phosphorus, potassium, calcium. These are the minerals. Example, in the pinus and oak you can find ectotrophic mycorrhizae. Now coming to endotrophic, the hyphae of fungus penetrates into cortex of the root. Here usually they play major role in phosphorus nutrition. So again coming to their example, orchids, coffee and some woody plants. So these type of endotrophic mycorrhizae, they are also known as VAM, that stands for vesicular arbuscular mycorrhizae. So as this fungus penetrates to the cortex, these corticals swell up to form vesicles, hence they are called as vesicular. The corticals swell to form vesicles. Next, here we have about cyanobacteria. They are autotrophic, that is they can synthesize their own food, and main point, they are widely distributed in aquatic and terrestrial environments. They fix atmospheric nitrogen. Examples are Anabina, nostoc and oscillatoria. So this is an important point. In paddy fields or the rice fields, cyanobacteria play a major role as biofertilizers. The blue-green algae increases soil fertility.